Hello everyone, my name is Adam Leszczynski, and I'm an incoming chemical biology graduate student at Stevens Institute of Technology in Hoboken, New Jersey. If you are considering being a part of this amazing experience, then I am glad to share with you my journey in research during the 2024 Japan IRES. Being a part of this program has been one of the greatest experiences of my life, and I will never forget the memories that I made in Japan, both as a scientist and as a resident. My story begins during the orientation week. When I first arrived in Japan, I was nervous because I had never traveled or even lived alone before. But with Mika and Carlos's help, I was quickly able to adjust to my new environment and learn what living in Japan was all about. I was able to learn where to go shopping, whether it be at a Family Mart or Aeon Mall, hear from the head researchers at the different institutions about their fields of study, and see for myself how easy it is to get around quickly and efficiently. By the end of the first week, I was not only able to take care of myself, but also make travel goals and realize them. In the first week alone, I got to see historical sites around Okazaki, as well as interesting landmarks around both Nagoya and Osaka. Research began during my second full week in Japan, where my PI at NIBB was Dr. Yasuhiro Kamei, and if you are a big fan of microscopes and home-cooked meals, then this lab is the perfect place for you. Over the course of 8 weeks, I was able to learn all about and eventually use spinning disc confocal microscopes, fluorescent scanning laser microscopes, atomic force microscopes, an X-ray micro-CT scanner, a scanning electron microscope, and a light sheet microscope. Because of the importance of microscopy to my work, my first 2 weeks working at NIBB were dedicated to learning the fundamentals of optics, and I even participated in a seminar where I built a simple microscope piece by piece to really understand what is going on at each step in the path of light through a microscope. Every day around noon, Dr. Kamei and his staff would prepare lunch for the people working in his lab, and I was able to watch and learn about his cooking techniques, and learn a lot about Japanese culture too. I even learned how to bake a fish. Along with learning about microscopes, I was able to participate in several different projects and learn to use a wide variety of techniques at NIBB. Many of these projects involved gene editing and a lot of work with plasmids. My first project was the construction of a plasmid that contained a gene for heat shock response factors, a fluorescent protein that was either M neon green or M cherry, and a gene for resistance to the antibiotic hygromycin. For 2 weeks, I learned how to design plasmids in the APE plasmid editing software, use restriction enzymes and gel electrophoresis to selectively cut and extract the genes I wanted to use, ligate my target gene to a plasmid backbone, use PCR to amplify the plasmids, and finally insert those edited plasmids into E. coli. After this project, I then moved on to a really cool experiment where the goal was to create glass slides whose surfaces were coated in the extracellular matrix protein laminin, which can be used to culture cells in an environment similar to the human extracellular matrix. I got to see something I had never expected, which is a chemical reaction that formed a covalent bond between glass and acrylamide, thanks to a silanization reaction that coated the outside surface of the glass with organic vinyl groups. After making the gel plates, I learned how to treat them with the cross-linking reagent known as sulfosampa, and then finally bind the laminin to the slide surface. One of the biggest projects I got to do had to be the experiment where I had the honor of working with Dr. Kamei's IR Lego system. By directing an infrared laser at a medaka embryo, the cells located where the laser was focused could be heated. The increased temperature of the cells would trigger the heat shock response factor proteins, which bind to the DNA to act as transcription factors for several DNA sequences that were genetically modified to activate as a result of the heat shock response. This would allow the selective activation of specific sites in the body of the medaka to express genes for fluorescent proteins. Between all of these projects, I was also able to learn how to work with the medaka, This included feeding them, identifying gender and phenotype, learning how to handle them for an experiment, and even how to dissect one. During my off time from working at NIBB, I would do my best to try new things and travel as much as I could. I enjoyed spending time at malls, convenience stores, and food courts in Okazaki, and I loved to take the bus and train to nearby landmarks and tourist attractions. On the weekends, I would travel by bullet train to a new city almost every week, going at 300 kilometers an hour through the Japanese countryside. I got to see Nagoya, Osaka, Kyoto, Tokyo, and Hiroshima, and at each city I had an incredible experience learning about the different historical sites, like castles and shrines, with museums dedicated to every facet of Japan's history. Nature seemed to be everywhere in the big cities, which was perfect for a nature lover like me. Rooftop gardens were common in Tokyo, there were aquariums in Nagoya and Osaka, and Kyoto and Hiroshima were short train rides away from stunning views of the mountains and oceans. No two cities were quite the same, and my love for taking the bullet train made the long journeys a blast. In the end, by being a part of this program, I feel like I've taken huge leaps as a researcher and global citizen. I think being able to experience a new point of view on research has allowed me to approach problems with a deeper perspective and become more appreciative of the scope of science and all of the ways to do research. By taking part in this program, not only do you improve in terms of technical experience, you also take home invaluable lessons about collaboration and what it really means to learn about another way of living. I would like to thank Dr. Mika Mutakata and Dr. Carlos Molina, as well as Dr. Yasuhiro Kamei and his wonderful staff at the National Institute of Basic Biology for organizing this incredible experience and helping me to have the greatest time possible in Japan. I would also like to thank the NSF for sponsoring this program.